Hello everyone, welcome to the Twisted DNA. Today we're going to talk about polymerase chain reaction, or PCR in short. Now let's start by defining what PCR is. So PCR is a laboratory technique that is used to amplify the DNA sequences. So what that means is that if you have a very small starting amount of DNA sample, we can actually use PCR technique to generate thousands to billions of copies of that particular DNA sample you have. And this revolutionary technology was developed in 1983 by the American biochemist Gary Mullis, for which he was also awarded the Nobel Prize in Chemistry in 1993. Now, let's look at the requirements of PCR and how the PCR works. So, for every PCR reaction, there are 5 core requirements, which are template DNA, which is the DNA sample that you have that needs to be amplified. Second is the primer. So, primers are the short DNA fragments, which are about 20 to 30 base pairs long, and primers are required to initiate the PCR reactions. Third is DNA nucleotide. They are our ATGCs, and these nucleotides are the building blocks of DNA that are required to construct the new strands of DNA. Fourth, we have is the Taq polymerase, which is a special type of enzyme that is used in PCR, and these polymerase adds new DNA bases. Final requirement is the PCR buffer, which creates the right reaction condition for the PCR. So once you have everything you need, you mix all the components in a PCR tube and load those tubes in a PCR machine, which is known as a thermocycler. So for every PCR reaction, there are 3 main stages. First is the denaturing, where the double-stranded template DNA gets separated into 2 single strands. The second step is annealing. So this stage enables DNA primers to attach to the template DNA. And third is the extension step, where a new strand of DNA is made. So the chart on the right shows that each of these stages requires a different temperature. Now let's dive a little deep in each of these stages. So first we have the denaturing step. In this step, the temperature is raised to 95 degrees Celsius, and at such high temperature, the hydrogen bonds between the bases of the template DNA breaks. So this allows the 2 DNA strands to separate. Now we have 2 single strands of DNA, where each of these strands will now serve as a template DNA. So denaturing step usually takes about 30 seconds to a minute. The second step is annealing step. So the annealing step usually takes about 30 seconds, and in this step the temperature is now cooled to 50 to 65 degrees Celsius, which enables the primers to attach to our target location on the template DNA. Since PCR primers are also short DNA fragments, they have to be complementary to the target sequence. Then the primers can only attach to the template DNA by a hydrogen bond. So these primers, like I said before, serves as the starting point for DNA synthesis. The final step of the PCR is the extension step. So here the temperature is increased to 72 degrees Celsius, and once the primers has bound to the template DNA, the polymerase enzyme can then attach and start making new complementary strand of DNA. The 2 separated strands of DNA are complementary and they run in opposite direction. So, in order to synthesize new strands of DNA, we need to have both forward and reverse primer. The Taq DNA polymerase is a special type of enzyme, like I mentioned before, that adds DNA bases and extends the DNA strands. So, why is this Taq DNA polymerase so special? Is because they are derived from heat loving bacteria called Thermus aquaticus. So, this bacteria is a thermophile and it lives in hot springs. So, the DNA polymerase that has been isolated from other organisms cannot tolerate such high temperature. So, we need such special polymerase enzymes for the PCR. After each PCR cycle, a newly synthesized DNA fragment also serves as a template to which the DNA polymerase enzyme can attach and can make more DNA. So these three steps of PCR reactions are repeated for multiple cycles to generate billions of copies of DNA. So there you have it, and that's how the PCR reaction works. And that's it for this video. I hope you like it. If you do, please don't forget to like, comment, share, and subscribe. And I'll see you all next time.